joining us from online, perhaps this morning. Yep, I see him there. And Xiaoming Zheng is also an online organizer. Thank you for joining us. So this workshop has a couple of purposes. We're very excited to hit two birds with one stone here. First of all, it's about mathematical oncology and looking at the interface between mathematical oncology, computational fluids, which many of you are in, and computational physics in general, and how they come together in cancer biology. The other important purpose for this meeting is to celebrate John Lonegrub's birthday. So happy birthday in advance, and also behind John, and thanks for joining us to celebrate. Our first our first talk here is by Thomas Hillen, who's going to kick off the series here. He is so famous, he's infamous. It doesn't really need an introduction, but he's been a good friend for many of us for many years and an absolute leader in the field of mathematical oncology. And I'd rather spend less time introducing, more time to him for speaking. So thank you for joining us, and then we'll do Q&A at the end.
Yeah, it was. So now it's better. So typically, for example, in my lab, a student would get a problem, devise a model, analyze the model, maybe related to data, and draw conclusions. But John did it differently. He built a modeling machine. Over many, many years, students then took the previous model and then extended it by including tissue stiffness, or including immune response, or including negative feedback, et cetera, et cetera. So it really grew into like a modeling machine, right? And then of course, this is now a very, very powerful tool where it can do lots of things. And I'm sure we will see some of the results here in this meeting during the week. So great to have you here. Now coming to my talk, John, you also worked on glioma modeling, this wonderful work with Jana Lipkova. And I will tell you in my talk that everything you did is wrong. Okay, maybe I shouldn't say it this way, but I will kind of. So my story goes, I will kind of motivate that maybe we, including me, are using wrong ideas, but then I kind of make the loop and then show later that it's maybe not so wrong at all. So, okay. This is about microtube-driven invasion of glioma. And before I start, let me get the co-authors. So that's the thing, yeah. So this is based on 3 papers. So the first one is with Nadia Loy, Kevin Painter, and Ryan Thiessen, where we derive the model for this microtube-driven invasion. And I'll tell you in a second what that is. With Alexandra Shinta, we looked at a specific experiment where they had a wound healing assay where they wound the cancer. So they're not wounding the patient, they wound the cancer and see how the cancer regrows. It's quite interesting. And then with Ryan, we worked on a more theoretical approach to look for traveling invasion waves, also because there are really interesting mathematical problems showing up in this work. Okay. So modeling of glioma, many of us have done this ourselves. And we certainly know that this is a very hot topic and has been done essentially starting with Christine Swanson about 25 years ago with her proliferation infiltration model. And so here are typical examples of glioma. And what you see immediately is a diffuse character, right? So these are not lumps of tissue which you easily identify as a white spot and then take out. These are highly diffuse, in particular here, for example, it's very difficult to find out where's the boundary of the tumor and how far should you treat or how far should you use irradiation, etc. So then seeing these pictures, it is a natural assumption to assume that some diffusion process is at work. And so the standard models for glioma, they're all based on an idea of diffusion. And here's some work on this. So as I mentioned, the proliferation infiltration model of Christine. Russ Rockner and John also in your model, this shows up. And there are lots of papers and lots of people who are using this type of approach. Another class are go or grow models, which have been also studied a lot with a lot of people. So the idea here is that you divide the cancer population in 2 bits. One population is moving and the other part is proliferating, and they can switch between the states, but they don't do it simultaneously. So they either move or they proliferate. And these models have been proposed for glioma and have been used with some success as well. And then a class of models I was involved in are anisotropic models where we use diffusion tensor imaging. And we have learned that glioma cells move along white matter fibers, for example. They use oriented movement using the fibers connecting the brain tissue. And there are a few work on this anisotropic diffusion as well. So what I'm going to show you next is that all these approaches are wrong. And because at this stage, when we did all this modeling, even in 2017 or so, we really did not know how do glioma cells actually move, how do they invade. There were assumptions, it was clear like from the population growth that there is a tendency following blood vessels, following white matter fibers, but how they move microscopically has largely been unknown until the work of Oswald. And this is what really brought all this work into start. So Oswald in Nature in 2015. So of course it took a while before it trickles into the more theoretical realm. So we didn't see this in 2015, we saw it later. Anyway, so this is, these are experiments on living mice in the brain of living mice. They found a method to videotape the growth of a cancer in a living mouse. And what you see here in green are the glioma cells, the brain tumor, and in red are blood vessels. And then in between is also healthy tissue, which is just not mapped here. And what you see is a very characteristic kind of almost a network formation. So this is one glioma cell. It sends out a very long protrusion. These are called the microtubes. Here's a glioma cell sending out a very long protrusion, and so on. And then here in the back, they seem to form a network even, connected with these microtubes. And Jun, I know you worked on micronemes, so this is probably maybe a similar process, just a different name. Okay. So I want to show you this in a video, so you can get a better impression how this works. And here I have 3 videos. Now Shane told me how to do this, so this, oh cool, there it is. Do you want to shed the light? No, I think this is okay. Let's get this started. So you see from the top the glioma growing into the healthy brain tissue, and they're sending out these microtubes, and then they grow along these network, and then they form a network in the back. Okay, and then here I think they show one of the individual microtubes. I stop it. Okay, so you see in the white box, can you see it? Right, so they're like 2 little tips, and you will see one advances and one retracts. And the authors say this is not a split. These are 2 microtubes which are optically behind each other. Yes?
Each cluster, each green cluster on the left,
Mm-hmm.
and quite a thin one, like
Yeah.
in the rectangle. Is there a reason, like for density of cells?
Well, the cluster on the left is a cell. It's a fully functional cell. And the line is just a microtube. So it's an extension of the cell. Yeah. So let's run this further. So you see one advances, one retracts. So there's some dynamic going on. They're relatively straight, not too random. Okay. And so then once this process has happened, here's a Z stack. So where you go through a tumor tissue stack by stack. And then you see how this network has formed. Is it running? No, I need to hit play. Yeah, so it's going through this tissue, and you see the cells, and you see also the network which has formed very clearly. So it almost looks like actually brain tissue and not cancer tissue. And then this has the fascinating effect that the cancer cells can relay signals along this network. They relay calcium signaling along this network. And if you see this, well, I'll show you in the next video, actually. do this here. So here you see calcium signaling inside a glioma network. You see it just lighting up certain parts of the tumor. And when I saw this the first time, I was completely shocked. I mean, this is essentially a thinking tumor, right? So it's like it tries to do the same thing as brain tissue, tries to signal and be connected. And the authors also have shown that this actually helps the cancer to repair wounds or to repair damage. So even if you cause damage through radiation treatment, say, this microtube network can actually help to get this fixed. Very crazy. Okay. So where's my talk? There.
It was more random.
Hmm?
Last year, it was more random.
More random, what do you mean?
I mean that there was no specific driver.
Exactly, yeah. So it seems like there are pacemakers which just light up randomly. And then this. So is Shane coming to rescue me? Thank you.
No worries.
There we go. Thank you very much. And this is the only video, so I don't have to do this again. Okay, so here's the outline of my talk. We started a bit late, but I want to be done by 9 o'clock, say. So we have to see how far we get. What do I want to do? I want to first derive the model, which is work with Nadia, Loy, Kevin Painter, and Ryan. And then look at scaling limits and make relations to existing models. So I think this is an important result, which I certainly like to show. And we have a result on traveling waves with Ryan. Then I can talk about the wound healing assay with Alexandra, and then talk about applications to glioma, which is probably very interesting. So I might skip maybe 3 or 4 depending on time, but I certainly want to do 5. Okay. So tumor microtubes. 93% of astrocytomas have them, less so for oligodendrogliomas, but astrocytomas are the primary glioblastoma which we are concerned about. And so they are very, very prevalent in all these cancers. The tips extend and also retract. They follow directional features of the brain tissue, such as blood vessels and nerve fibers. They divide separately. And then the glioma nuclei, they divide, but the cell doesn't divide. So then you have a cell with 2 nuclei. And one nucleus is transported along a microtube and settles somewhere. And then it forms a new bulk cell wherever it has settled. Okay, so they move along to form new glioma cells. And a glioma tumor microtube network forms, which communicates via calcium waves. So there's an earlier paper by Martina Conte et al. 2022, where they look at the microtube extension in a little bit more detail, including mechanical principles there. Okay. So our model here has 5 ingredients then. So we thought about this a lot. I mean, this work took like 3 years. And first you need to decide what are the major players actually, what do you want to model? So we decided actually to use an approach which was made popular by Leah Keshet on fungal growth. Right, fungi grow and they lay down hyphae and they form a network as well. So we mimic this. And in this framework, you identify a tip, which are these little red arrows here, microtube tips, and then the tips advance and they lay down a microtube as they advance, which is M. And then we have tumor bulk, which is just the bulk cells here. We have moving nuclei, which move along the microtubes. And then we have resting nuclei, so they settle somewhere and then they form new cells. Okay, so this is directed motion, right? The tips have an arrow, so this is directed motion. This is not a diffusion process, or at least not yet. So what kind of model do you use? Yeah, you have a question. It is just essentially a delay. So the nuclei rest and then they build a new bulk cell. It takes some time to do so. Yeah. So what kind of model for this directional movement? Well, asking Thomas Hillen, who has worked on transport equation all his life, maybe we should use transport equations. Okay. So that's what we do. So let me walk you through these 5 equations. It's a little bit painful, but I mean, it's a complicated process. So we have to look at this. Okay, so we use the transport equation. So P is the tip density, depends on the velocity. And so then the left-hand side is just the characteristic derivative in direction V. So describing the movement in direction V. V is a unit vector, S is the speed. And then the change of direction is described by an integral operator L. And I don't even write out L. So there's lots of publications of how this can be done. It's in the appendix of our paper where we can look it up. But this distracts from the story. So L is an integral operator describing directional changes. Then they just decay. Anastomosis is if they find a new, if they find a glioma cell, they make a connection. And then they are no longer tips. And then there's a production term from the bulk. So the bulk cells produce microtubes up to a certain carrying capacity. So we assume a logistic term here. And then, as I said, this is based on Lea Keshet's work on fungal growth. It's kind of the same mechanism. And these things I explained. Yeah. Oh yeah, this psi here for the experts. The psi is an equilibrium distribution of the turning kernel L. And this shows up here so that the new microtube tips choose the correct directions in this environment. Okay. So then they lay down tumor microtubes. So then the tips lay down the microtubes to this term, and they can mature into bulk cells if they interact with a nucleus. Then the bulk is essentially stationary. They don't move, so there's no diffusion term. They have a logistic growth term, and they grow on their own. That's the first bit. But they're also produced by the resting nuclei, which is the second term here. And then the moving nuclei, well, they also have a transport equation because they follow the microtubes. So then the turning kernel here, it's written out in a little bit, where little m is the mean direction of the tumor, no, it's the normalized direction of the tumor microtubes. So this means that the nuclei follow the existing microtubes. Okay, and then they grow and they mature into bulk cells. And then we have the resting nuclei, which is a simple equation. From the interaction of nuclei and microtubes, they are produced and then they mature into bulk. So put them all together. Here is the equation again. And this looks rather complicated, right? So I said, we took a while to come up with this model. It was quite some effort until we were happy with all the terms, but I think this is a good choice. Good. So what do we do with this? Right. So we cannot go to the doctors and say, hey, you just use this model. And so we have to do something with it. And the first idea is to just look at the kinetic part. Forget all the space, right? And just look at the kinetic part, get an ODE and see what this does. So we do this here. Well, not yet. These are the parameters. Now, since Oswald has all these videos, and I've shown you 3, but there are more videos, we can actually measure the speed of invasion of the microtubes. We can measure the speed of the nuclei in the microtubes. We can see how many microtubes per cell. We can see how many cells per square or per cubic centimeter, etc. So we can get all these units from the videos. So this was also some interesting work. But I think we have a pretty good handle on these parameters. Okay. So now let's look at the kinetic part. So this is the kinetic part, lots of logistic terms, etc., and just see what it does. So we have continua of steady states. We have a continuum of disease-free steady states. So M1 would be the microtubes, but then there's no cancer and no nothing else. Or we have a disease steady state where K is the cancer at carrying capacity, and then surrounded by some microtubes as well. Okay. In the simulation, we can see better what's going on. So here, what do we have over time in hours? Here are the densities of the 5 ingredients. So you see here at the very beginning, it all starts with the tips. The tips in red are coming out. Okay, and then they are followed by the tumor microtubes in blue very quickly. And then they guide essentially the tumor bulk in black. coming out and growing. And the moving nuclei and the resting nuclei, they have lower densities. They kind of facilitate in the process, but they do not accumulate. They will just be cycled through. But very interesting. So the tumor microtube dynamic leads this growth, and then the black one follows after. Yeah.
Just a question, because all the maxima
Yeah.
are things at about the same time, like around 250 hours.
Yes.
Is there a reason?
There's a reason for sure. Yeah.
But we don't know.
I don't know. We would need to look at it in phase space, but they probably make their way around a steady state and come back or something. Yeah. And I'm pretty sure if you take the derivative of the black one, you get the same maximum as well. Yeah. Inflection point. Yeah. Yeah.
Because one of these is leading and the other.
Yeah. Right. One is leading and the others follow. Yeah. There's no change in the system, right? There's no change in the parameters or anything. It's just the dynamics which does this. Yeah. But we can change the system by doing like a surgery. I mean, if you have an ODE, you can just play around, and so we just take out the tumor and just see what happens at 3,500 hours. Yeah. Right. So here at the green line, we do the surgery. We set black to 0. We keep everything else. And you see it comes back super, super fast. So meaning if you take out the tumor cells, but you forget the microtubes, then there's lots of material to grow back. And this is, I think, what's happening actually in glioma treatment a lot, because the microtubes will not show up in MRI imaging, right? And then if you take out the tumor bulk, but if you leave this microtube network behind, then they have material to grow back very, very quickly. Back to the full model. So Nadia developed actually a code for the full model of 2 transport equations and coupled to 3 other equations, and solved it. And we get a nice invasion fronts coming through. These are all the 5 ingredients. Again, it is led by the red and blue. So the tumor microtube leads the invasion front very clearly here as well. Okay. Oh yeah. So let's look at the model reduction. So this, I find, is a really exciting story, because we are now here. So the full model is the black one here on the left corner. And now I like to trickle through a whole bunch of simplifications and scaling assumptions and fast and slow dynamics and all that stuff. Assumptions A1 to A6, until I get to something which is called the bulk isotropic model. So this doesn't tell you much, but if I overlay this one here, it tells you a lot. So the bulk isotropic model down here is actually Swanson's model. Swanson, Rockner, etc. The model I said John used also in his work. So this shows up after a series of simplifying assumptions. And this is really cool, right? Because now you get an a posteriori motivation of the earlier models. And similar for the go or grow. So this one here, Farm Hatsukiro, is the go or grow formulation. And also similar for the anisotropic model, which is here, which was derived by us and Soholescu and others. So these models all show up after a series of simplifications. So let's look at those. So A1, A2. So we assume the essentially the nuclei dynamics is fast. So resting nuclei are quasi-equilibrium. Microtubes are assumed to be quasi-equilibrium falling in the tips. And so if we do this, I said these 3 equations equal to 0. I assume these are in quasi-equilibrium. And you see there's a recurring term, kappa nm, kappa nm. Kappa nm bar means the integrated version over velocity space. But anyways, right, so you can simplify this. Kappa nm is sigma p, you put it here, and then r you put there, etc. So it's relatively easy to do that simplification. And so then we get what I call the tips bulk transport equation. So the tips in P and the bulk in B, and it's still this transport term here, the kinetic term and the equations. Okay. So then deal with the kinetic terms. I did this a lot of times in my career using parabolic scaling. So we scale time by epsilon square. We scale space by epsilon. Epsilon is a small parameter. And I assume the reaction terms scale on the slow time scale. And then this becomes a diffusion term in leading order. So I call the leading order term U. So P0 is a leading order term in an expansion, and then bar means integrated on velocity space. And so then this U satisfies like an anisotropic diffusion term here, and the interaction terms here and here. And this anisotropic diffusion term can be actually computed by the second moment of the equilibrium distribution of the turning kernel L. So of course, there's a lot of theory behind this, D and putting it there, and the psi, etc. Again, I skip the details, but I'm happy to talk about this later if you're interested. But this is nice. This is getting really nice. So now we have a reaction diffusion model coupled to an ODE. So it seems to get somewhere. An important special case is isotropic diffusion. So in many cases, the diffusion tensor becomes the identity matrix multiplied by a constant, which is then the diffusion coefficient. And then we get something even simpler. We get a model for the tips and for the bulk. Okay. So now take down your Marthy hat and put up your modeling hat. And look at this a little bit. So we have a moving compartment and we have a non-moving compartment. And the non-moving compartment reproduces a logistic term here. It also reproduces U. So this is essentially a go or grow model, right? You have a going compartment and a growing compartment. And so here we have a go or grow model, which I mentioned earlier already. Wonderful. Okay. And yeah. As I just said, and these are authors who have used the go or grow approach. And then finally, in the videos we see, we have so many microtubes per bulk, essentially about 3 or 4. So you can find a proportionality factor that the number of tips is proportional to the number of bulk cells. And then you reduce it to one equation. In the anisotropic case, you keep the diffusion tensor. In the isotropic case, oh wait. Okay, in the anisotropic case, then you get the anisotropic model, which I worked on with Painter and Swan and others. In the isotropic case, the diffusion term is simpler. You get the same terms otherwise. And then this is the model of Swanson. This is the proliferation infiltration model. But even more so, right? It is a Fisher KPP equation, and you can identify the parameters d hat, mu hat, k hat by the original parameters of the microtube network formation. So this is really cool, right? So we have these parameters from the experiments of Oswald, so we can actually compute what these hat parameters are as well. Nice. So what did we do? So that is the result of this scaling analysis. So we know exactly what these scalings are. They're written down here again. And we find the go or grow, we find the PI, and we find the anisotropic model. And I think this is a really nice result because now scientists can go and say, hey, look at these assumptions, which are satisfied and which are not. And then it tells you on which level in this graph you should actually be modeling, right? Or what data do we have? Do we have data on this and this and this? And so we can make a decision where to work on. Okay. So this is my rescue of your model, John. So as I said, this is a posteriori now justified and a good model as long as these assumptions are more or less satisfied. Very nice. Good. Okay, let's look at invasion speeds. Yeah, John.
So if you plug in those numbers, how close do you get to what Swanson and others have predicted those?
For the speed, for example, you're completely off. Yeah, that's what I wanted to show next. You're completely off in the speeds. So let's look at invasion speeds. Invasion speed is an important characteristic for you want to know how quickly a glioma invades into the tissue, of course. And so here we simulated all 5. How many do we have? How many models did we have? 1, 2, 3, 4, 5, 6. So we modeled all 6 for the invasion wave. I'm not gonna kind of tell you which is which. The interesting bit is you should see they all have different speeds. So here's one of the models, here's another one of the models, here's the full model. Another one has already run through all the way. So they invade at very different speeds, as you can see here. So this answers your question right there. And so here we measure the speeds, the full model 0.38 and the bulk diffusion model, this would be like the Swanson model 1.2. You can compute the theoretical wave speed, which is also 1.2. So they're not consistent at all. And this, we got very concerned about this. We thought, oh my goodness, this shouldn't happen. But then we thought, hey, we do scaling analysis, right? We transfer time scales, we change spatial scales, we do quasi-steady state assumptions. So all this messes up with the time scales. And if you mess up with the time scales, you mess up with the speed. So that's just the way it is. Okay. So for the Swanson model, for the proliferation infiltration model, you have Fisher KPP results, so you can directly compute the invasion speed, again, depending on the parameters, which is nice. Yeah, I skipped these details. And so then we know the speed here. Now we want to compute the speed for the go or grow formulation. And this is work I did with Ryan Thiessen, right here. And, excuse me, so here's the model again, now written in non-dimensional form. This is non-dimensionalized. And the problem here is interesting. This is a non-cooperative system. So for people who are experts in traveling waves of reaction diffusion type of equations, if it's cooperative, you can use the method of Xiaoqiang Zhao and others to actually estimate the wave speed. But if it's non-cooperative, you are screwed. So there are very little methods to do this. And so Ryan had to work really hard to find upper and lower solutions for this process. So if you find upper and lower solutions, you can squeeze your solution between those two and then get the invasion speed. So this happened here. So we get a kind of an implicit formula. So the infimum over rhos over this expression here is the minimum wave speed. And here's a simulation where the pink one is the upper solution, the pink and blue is the upper solution, and the black and red is the solution you are looking for. And so this matches really well, and this works fine. Okay. Yeah, so I think my time is really good. So let's look at the wound healing experiments. So now we wanted to see if our model with the tumor microtubes can actually do something useful and explain experiments. So here's experiments by Weil and Oswald. So this is the same group, the same Oswald as I showed you earlier. And there's tissue, again, in the mouse brain, and they take out a cylindrical region of tumor cells. And then you see the days, day 0, day 1, day 3, day 7, 14, 21, 28. And then you see regrowth in this region at around day 7, day 14, day 21, day 28. And it grows faster, or it grows bigger, denser than it was before. So you get kind of a wound healing for the tumor tissue, but you also get increased growth in this area here. So now we try to use our model to describe this these regrowth experiments. In particular, though, we want to know what the microtubes do in this case. And here is also from Weil's paper, an image of one glioma cell and the microtubes. Also here a little bit, they seem to extend into the void. They extend into the lesion, and then they give guidance for the tumor cells to come in there and grow as well. So, so then our model for this case is this one here. So here we have the lesion where the tumor cells were taken out. We have the tumor cells which form the network, and they form microtubes into the lesion. And then we also have healthy cells which can also grow into the lesion, but at a lower rate, yeah, because they are not cancerous. So that's our model. So our model now consists of glioblastoma cells with the microtubes and the healthy cells as well. So essentially meaning, since we are interested in the directed movement along the microtubes, we use the anisotropic formulation. So then this equation here will be used for the glioma, and this one will be used for the healthy cells, because the healthy cells have no microtubes and no preferred directions. So then the model is this. So glioma cells, this is again the term I showed you earlier as I derived the bulk anisotropic thing. Logistic growth with a competition term here, and then standard diffusion over here. Okay. And so now, model assumptions. We assume the domain is radially symmetric. Healthy cells move via isotropic diffusion, cancer cells via anisotropic diffusion. And now the radial symmetry is actually a strong assumption. But in this case, we can explicitly compute the diffusion tensor, because this has to be radial symmetric too. And this was done in a paper by Bika. So we use the formulation of Bika then for the diffusion tensor. And then we assume that the tumor microtubes provide a growth advantage. And this is part of our earlier paper as well. So where the parameters, you remember I had this mu hat, the growth rate, which depended on all the parameters. So the tumor microtube parameters are in there, and they increase the growth rate in the lesion region. But in addition, there's wound healing. It's a living mouse, so there's a wound healing mechanism, and wound healing mechanism increases the growth rate, right? It releases growth factors. The growth is increased, so there's a second effect there. Okay, and then these are simulations. So day 0, day 1, day 3, up to day 28, where we get regrow in the lesion, essentially in the same time scale as we saw in the experiments. Now this is completely homogeneous, right? We assumed radial symmetry. So in radial direction, it is completely homogeneous. But the time scales are right, and the densities are right as we get them here. So the simulations correspond to the experiments. And then while tested 2 treatments, one treatment with the tumor microtube inhibitor, and this showed reduced growth in the experiment and in our model. And they tested an immunosuppressant where the growth is only temporarily inhibited, which you also get with the model and they show with the experiments. So the main outcome here is we find that the dominant mechanism for cancer regrowth are the increased growth signaling through the wound healing and through the growth benefits of the tumor microtubes. And the oriented diffusion via radial guidance is not so important. It increases growth, but it is not the dominating effect. So I was a little bit disappointed. I thought this anisotropy from the microtubes is maybe a dominating factor, but it doesn't seem to be. It contributes, but it is not the most important factor. The most important factor are the regrowth signaling through the wound healing and also through the tumor microtubes. Okay, well, that's fine. What do I want to do now? Okay, yeah. So now let's look at where is this used in brain tumor modeling? How far are we with all these models to look at patients, which are non-mice, but maybe human patients? And let's start with the classical model by Swanson down here. So, so typical simulations of the Swanson model look like this. So in black here, there's a tumor, and then surrounding and level sets is a diffusion model or the diffusion process solved on the brain domain. And you can see the same tumor from different directions here. And this is a diagnosis, this is a death. And important here, it will have infiltrated much further than you actually think. So if you only treat the black laid out region, you will miss a whole bunch of tumor. And I think this is one of the major insights which came out of Swanson's model in this regard. Okay. Let's look at the go or grow model by Olivier Sort in France. So he did this simulation, but this is not a cancer patient. This is a healthy person where they scanned a healthy person and just put a tumor in a simulation, not in real life, and then let it go. And so then this is the growing compartment. This is the invading compartment here. And this is the necrotic core. So they also included an equation for the necrotic core, which I did not have. But these first two are go or grow, simulated, as I said, on a healthy patient, which is then not a patient by definition. Anyways, so okay. And then the anisotropic model, we used it with Amanda Swan for patients, for 10 patients. And here I show you maybe 2, I think, where we compare the anisotropic model and the Swanson's PI model. And what you see here in black is the medical rendered tumor. And in white is our simulation, which kind of predicts where the tumor should be in our model and in Swanson's model. Now the colors from blue to red shows the level of anisotropy. So dark red stripes mean white matter fibers, and blue means more or less homogeneous tissue. And you see both models do equally well. So I think the white line and the black lines in both situations agree nicely. So this is nice. Here's another case where it's a little bit different. Patient 7. So in Swanson's model, you get kind of a little bit more growth in this direction, which we don't have in the anisotropic model. So a little bit can be gained by using the anisotropy of the brain. All right. And then here's maybe another example. All right. So here is an outgrow in the top, which is completely missed in the Swanson model, which we do a little bit better, but there's still a bit which is also missed. So it's certainly not the end of the story. Okay. Yeah. So then if you look at patients, so the PI model has been used for hundreds of patients. The anisotropic model has been used for 10 patients. The go or grow model has been used for 1 healthy person. And that's where we are at the moment. Okay, so there's still a long way to go to get all these details into patient modeling and maybe patient-specific results. Okay, so this is lots of information. So I think I can wrap up. Yeah, so the tumor microtubes drive glioma. The tumor microtube-driven glioma model can be reduced to the previous models in special cases, which is great. It opens the door for more targeted patient treatments. It is mathematically really interesting, but there's lots of questions I did not touch and which could be touched. And the anisotropy is not the dominating factor in the wound healing assay, which I found interesting. Great, so that's the end of my talk. Thank you very much.
Are there any questions? I'm going to pass the microphone to anyone who has questions so the people online can also hear them. Thank you so much, Thomas, for this great talk. John.
I'll ask another one. So in the kind of bulk equation, so either the anisotropic or the isotropic versions, the tumors that you predict, and we do the same, they tend to be much more compact geometrically than what's observed. And so have you guys tried to use your model in non-symmetric cases to see if that might be different with these?
For compact, you mean it's more packed together?
It's more spherical than what you would observe. And you showed an example like that towards the end.
I'm still not clear. I understand what you mean.
So if you go back a couple of slides.
Okay.
No, sorry, too far.
Too far? Okay.
Yeah, so for one of your patients, not this one, not this one, that one.
That's the worst
so you
one.
See, So you see how they're, at least the doctors think that it's invaded
right here, right?
Yeah, so it's more less spherical. The model is predicting more spherical, both in the isotropic and anisotropic case. So my question is, with the microtubules help that? So if you tried to simulate, I don't know if you have tried to simulate the non-symmetric versions of your equations, whether you might see features like that, which you don't see in the classical.
So we thought about this case as well. We came to the conclusion that this is pressure, because it is right next to the skull. Right, here's the skull. So there's actually pressure buildup and deformations. And so we have to become mechanical. We need to include mechanics to do this. So this is our suspicion. Yeah.
We have a question from Kathleen.
Thank you, Thomas. I was wondering about the wound healing stuff that you did,
Mhm.
and if you thought about why it regrows so fast. What does the model tell you about why it regrows so fast? Because presumably in the model now there's like a void where the surgery happens, so there's no structure at all. So what does the model tell you about why it regrows so fast and dense in the void when there's no structure there?
Yeah. So the structure there will be the immune response, which is not mapped or not measured in these experimental data, but there will be immune cells and fibroblasts and everything around, and they release growth signaling. And so we just assume that the growth signaling in the lesion is higher, and the carrying capacity in the lesion is also higher based on that. But since we don't have detailed information about the immune response, we couldn't be more detailed there.
Great. Are there any questions online? I think go ahead and just unmute yourself if you have one.
Thank you. Thomas, just for curiosity, you spoke about glioma cancer cells transmitting information in between them.
Right.
Was it detected for other kinds of cancer cells and for other organs?
I don't know.
Is it just
Yeah.
is it for now just a unique phenomenon for glioma-related cancer cells?
Okay, this is a question to my more like my philosophy or my understanding of cancer. I think the cancer in a tissue uses actually uses mechanisms which are typical for that particular tissue. So I think a liver cancer tries to mimic liver function, and a brain cancer tries to mimic brain function, etc. So I would think it's typical for brain tumors.
Xiaoming, I see you're unmuted. Do you have a question? Well, I think you've stunned everyone into silence with this great work. So if there are no other questions, I'm sorry. So if there are no other questions, let's thank Thomas again for a wonderful introductory talk. Let's kick us off here today.
Thank you.
We're now scheduled for a coffee break and be back here at 10:30. Is the coffee out here or upstairs? Oh, just directly outside here. Thank you, everyone.